Why is the Human Genome Project an important milestone in the study of biology? So, how did the Human Genome Project come about? With advances in human genetics being understood at a molecular level, scientists came together in the 1980s in an attempt to sequence all base pairs which make up the human DNA. The Human Genome Project is aimed ultimately map all genes collectively known as genomes. The Human Genome Project started in 1990 with collaborative efforts by the National Institute of Health, US Department of Energy and research groups from UK, Japan, France, and China. American genetic scientist Francis Collin initiated the program, and more scientists quickly joined him globally. Many technological advances in genetic sequencing improved the efficiency of Human Genome Project at the time, including better tools and instruments to decipher the base pairs in DNA, improved computing capacities to manage the data obtained, and most importantly, the use of Sanger's sequencing, developed in 1977 by Frederick Sanger, when the automated method of sequencing hundreds of base pairs in DNA at a time allowed the project to be completed ahead of schedule. The first draft in Human Genome was published in 2001, thereafter completed in 2003. The base pair sequence of entire human genome was mapped out, and it was revealed that humans approximately had 25,000 to 30,000 genes. That's crazy. Techniques used and invented to make HGP, Human Genome Project, including recombinant DNA. The Human Genome Project used several techniques to map the human genome. The DNA sequencing is determined the order of nucleotides, base pairs in a segment of DNA. The main sequencing DNA techniques used in the Human Genome Project: 1. Recombinant DNA technology. Recombinant DNA involves isolating one gene or segment of DNA and combining them with other vector cell DNA for replication. Commonly used vectors are viruses, bacteria, yeast cells. With this allows for DNA cloning, increases in quantity, which allows large amounts of DNA for analyzing nucleotides-based sequence. 2. Polymerase chain reactions, PCR. Using PCR, highly specific segments of DNA can be rapidly increased in number. PCR involves separating double strands of DNA molecule by heating and cooling the molecule with a polymer enzyme. This results in DNA fragments amplifying in large quantities within a span of minutes. Once the DNA is amplified, they are sequenced using the Sanger sequencing method. This DNA is replicated through dideoxy chain terminators, which produces short DNA strands which are ordered by size, short to long, to show whole DNA sequence. Discuss the benefits of Human Genome Project. Many different benefits have been gained by understanding the complete genetic sequence of human genomes. Via the Human Genome Project, main benefits include biomedical research. By identifying differences in genomes maps it can help identify the genes responsible for heredity, diseases such as muscular dystrophy, retinoblastoma, Fragile X syndrome, and more. This can help find the reason behind diseases and likelihood of inheriting them, and treatments. Risk factors, as some people are genetically more prone to have reactions to certain toxic chemicals. The Human Genome Project can work out the likelihood of how much damage to health can occur, such as the radiation. Forensic science, using DNA sequencing and matching as evidence to find criminals. The gene therapy, possible changing and replacing the faulty genes that have been identified in the gene sequencing in some diseases. So, what are linkage-based maps? Genetic linkage-based maps. Genetic linkage is when there are two different forms of genes or alleles present in the same chromosome. Recombination is when two genes of homologous chromosomes cross over, which during meiosis 1 is a new combination. When two genes are closer together on the chromosome, they cross over less and so they are genetically linked. When genes are far apart, they are more likely to undergo recombination, separating hence by working out how frequently two genes are inherited together. Maps can be made based on relative distance between them on the chromosome. So, how genetic linkage maps are used to identify positions of a gene on a chromosome. Genetic linkage maps are included the distance between the genes to put into map units. One map unit equals 1% chance in recombination between two linked alleles. Overall, genetic linkages can be used to find the exact location of a gene on a chromosome. Reasons why maps couldn't be used to map the human genome. The Human Genome Project could not rely on the genetic linkage map. The sequence of the DNA, the genetic linkage maps only provide relative locations of specific DNA markers. The Human Genome Project aimed for specific understanding of linkage maps also rely on finding phenotypes, which are inherited by humans, and this could not be done for DNA samples used in the Human Genome Project based on ethical reasons. Mapping cannot work out the order and the position of relations to other genes, a chromosome in terms of recombination frequency. Human genetic linkage maps are based on rates of recombination across the genome. These rates in humans vary by sex and parents, for whom alleles inherited by chromosomal position, and by genetic features such as GC content and repeat density. Gene mapping determines the position and the order of gene relative to other gene along the chromosome. A well-marked linkage group extends from marker located at one end to the chromosome to those in the middle, and the markers located at the other end. The number of linkage groups in an organism is equal to its number of homologous chromosome pairs.